Welcome again, for those of you who are here this morning. When you see the media talking about evolution, it's often presented as if it's a fact, and you're an absolute fool if you don't believe it. For example, Ernst Mayr, who was a prominent evolutionist, he says, no educated person any longer questions the validity of the so-called theory of evolution, which we now know to be a simple fact. Likewise, most of Darwin's particular theses have been fully confirmed, such as that of common descent, the gradualism of evolution, and the explanatory of theory of natural selection. So here is a clear statement from an evolutionist that we're to accept it. It is fact, and that's the way it is. So, another claim here, such claims that evolution is a fact. This was written by Michael Denton, and he is not a Christian, just to put this into context. He counters this. He says such claims that evolution is a fact no longer needing proof are simply nonsense. For Darwin's model of evolution is still very much a theory and still very much in doubt when it comes to macroevolutionary phenomena. Furthermore, being basically a theory of historical reconstruction, it's impossible to verify by experiment or direct observation, as is normal in science. And he makes a key point there, Michael Denton. If the evolution from molecules to man if it occurred at all, it has occurred in the distant past, because after 50 years of attempting to see it in the laboratory, they haven't seen it, and it isn't observable today. And so the conflict that we're talking about here is not a conflict between science and religion, or science and Christianity, if you want to put it that way. No, it's a conflict between two views of interpreting history, of what happened in the past. And so keep this in mind. It's not science versus creationism. It's two views of what happened in the past. And evolution is simply not derived from the evidence that's available today. It is imposed on the evidence. I mentioned this this morning, and how many weren't here this morning or didn't hear this morning? There were a few of you. Okay, I will take the time then to define evolution. The idea is that, and Ernst Mayr said this, but it comes from an original single cell, all the variety of life that we see around us. And genetic information has been increasing through time. I mentioned that the simplest cell that we know of today that can reproduce itself has maybe roughly 480 genes. And that's what it takes to make an entity that can reproduce itself, whereas the human genome has about 30,000 genes. So somewhere along the line in the theory of evolution, the idea is information has been increasing through time. At the top are all the species we see around us. Some species have died out, whether they died out in the flood or died out since the flood and gone extinct. That's the ones that haven't made it to the top of the tree. And then there are the missing links on the branches. And so Lynn Margulis, another prominent evolutionist, she said every form of life on Earth, oak tree and elephant, bird and bacterium, shares a common ancestry with every other form of life. And so plants, animals, and humans have all come from that original cell. That's the definition of evolution. So what difference does it make? Somebody could say, well, I believe in Jesus. Isn't that all I need? Maybe God used evolution as the means whereby he created the variety of life that we see. So why should we care about this? Why is this an important topic? So one reason is because God states that he created plants and animals by their kinds. He didn't create them from pre-existing entities. And the writer of Hebrews reinforces that in Hebrews 11:3. Another reason we should be concerned about understanding this is that mankind is presented as a unique creation. We read one account of the creation in Genesis 1, and then it's expanded in Genesis 2. And we see how God formed the body of Adam out of the dust of the earth, and he breathed a spirit life into that as a unique creation. And it wasn't that somewhere along the line a hominoid creature was endowed with rationality. The Bible presents it quite differently. But if mankind is a product of evolution, well, then what happens to Genesis 1? Genesis 1 can no longer be taken as a historical account. It is only a story. And if it's a story, then why should we take Genesis 6 and 8 to 8, the account of the flood, as having any reliability? And for that matter, why bother to believe in Abraham? And if we don't believe in Abraham, why believe in David? And if we don't believe in David, why believe in Christ? It's that simple. If any part of the Bible fails, the whole Bible fails. And we as thinking people should desire to know the truth. We should want to know what the truth is and how it's supported by evidence. I pointed out in one of the presentations this morning that 170 years of evolutionary thought is one of the major contributing factors to the rampant immorality we see around us today. And one of the primary reasons why we should care about this, and we really need to question evolution, is it's not accepted on evidence. And I'm going to show this to you. It is accepted because men want to believe anything other than that God is a creator. It really comes down to that. That's the reason why we should do it. And in the end, evolution is a form of idolatry. It's worshiping the creature rather than the creator. So let's look at this in a little more detail. Many times the opponents of God's special creation, they claim that what we believe in is that God created all the species. In fact, you'll hear people say, well, how did Noah take all the species into the ark? No, we don't believe that God created all the species. That is false, and some of those species have gone extinct. What God created was kinds, and the kinds in our modern taxonomy may represent the family level in our modern taxonomies. They may, above the species level. They certainly are. And in that kind, there's a broad spectrum of genetic information, and those kinds have adapted through natural selection. Yes, we accept natural selection. Also through artificial selection, where humans do breeding, and they have diversified into different ecological niches, and that is where the species have come from. So the modern species are the result of developments out of the genome that's available at that. And so there have been, since the flood, there have been points where there have been explosions of speciation. I'll give you an example. After a hurricane in the Caribbean, it might wipe out a huge amount of the life there, and so let's say the lizards are destroyed. And this is actually one of the good examples. There's real literature on the lizards that are destroyed on the island. After a while, some lizards make it to the island again. They float on water, plant rafts or something. They make it to the island. And all of a sudden, there's an explosion of species that appear because they go into different niches. So those migratory lizards take advantage of the availability of an island where there's no competition for their niches, and then they explode into that. And that happens over and over again in biological development. So species form from a broader genetic availability of information. But one of the things that you need to realize is when a species is isolated from the population, it loses genetic information. So take this as an animal kind that Noah would have had in the ark, and out of that, different species have developed from domestic cats. By the way, that's our cat. He thinks he owns the place in the middle. Now, you cannot breed a lion and a domestic cat. It doesn't work. However, there is basically a chain of breeding, like lions and tigers can interbreed, and you can work your way down the chain. And domestic cats can interbreed with lynxes and bobcats, for example. Doesn't necessarily produce a fertile offspring. But so there is this continuous network within the kind, but they have diversified to such an extent that they've lost the ability to interbreed now, and they have less genetic information because they have been isolated. And genetic diversity has actually declined, and there's been a loss of information. And I used this illustration this morning, that when you breed dogs, that's not natural selection, it's human selection of the breeds. The breeds actually become less robust over time. And if you inbreed too much, they explode. And I mentioned that probably the most, if you want a really robust dog, you want a mutt, because you bring all of the different gene threads back together. I had a friend who raised Great Danes, and they have a very short life expectancy relative to some other dogs because they have a heart condition, and so they don't survive very long. So this is the result of losing robustness in their genes. But even after 3,000 years of selective and extreme selective breeding made by humans, these are still dogs. They haven't changed from being dogs. So the mechanism that evolutionists need for, in their theory, to be successful is described here. The developing research area on how the vast biodiversity on Earth evolves accepts natural selection, and we creationists accept natural selection, by the way. That's not a problem, and I'll show you that in a minute. And 3 other established forces of evolution as its basis. These include mutation, random genetic drift, and gene flow. Now, I am not going to get into random genetic drift and gene flow today. That's too technical for this. I'm just going to deal with natural selection and mutation, because those are the 2 primary supposed engines of evolution. So let's look at natural selection. So you have an initial canine kind. We'll say it's a dog, so it's a species within the canine kind. And it has a fairly broad spectrum of genetic information available to it. And it has in its genes for long hair and genes for short hair. Picture that, right? A mother dog would give birth to pups. Some of them would have short hair and some long hair. So she has the genetic information for long hair and short hair. And then what happened, and you can see I'm using L for long and S for short. And by the way, this is a caricature, because often fur length is not usually associated with a single gene. There's multiple genes that contribute. Studies have been done. For instance, human skin color, there's at least 2 genes that interact, and they're quite a bit apart, and they interact for human skin color. So you can't find a single gene necessarily that deals with fur length. But keep it simple. So they have offsprings that can have a mix. For example, that one of their offspring, when you have both male and female dogs that have long and short genes, and the 2 sets of chromosomes come together, they can give birth to mix both genes, or some of them will have only long hair genes, and some will have only short hair genes. And so let's take the example that they give birth to those, and they're in a very cold climate during the Ice Age, for example, in the northern regions. And the long-furred dogs survive the best. And they give birth to, because they only have the long-haired gene, they give birth to only dogs with long hair. And over time, there's no more short hair genes available in that population pool. Now, they're still dogs, and they could breed with another type breed of dogs, and the genes could be reintroduced. So the canines have adapted through natural selection to their environment, but they have lost genetic information because the original pair had the genes for long hair and short hair. The one that is adapted to the cold climate and survived the best has only the genes for the long hair. And so information is lost when small breeding populations are isolated from the larger pool of genetic information. And this is exactly the opposite of what the evolutionists need, which is microbes to man. You need increasing genetic information, added genetic information. You don't need the loss of genetic information. And so this isn't evolution. Natural selection doesn't support evolution. It's taking already existing genetic information, and the animals or plants are more adaptive that have certain genes, and then if they're in an isolated breeding pool, they lose genetic information. Let me just, so that you can understand a little bit about what I mean by isolated breeding pools. My wife is from Puerto Rico, and there are certain peppers that have a bit of a spice to them, which we can't get in the supermarkets, not surprising. And so she and one of my sons, they got the seeds of a particular type of pepper that we wanted to grow this summer. My son Andrew, he grew them in his garden. My wife grew them in a flower box on the porch. So they were isolated, and they produced the same seeds the next time. But Andrew grew them along with other peppers in his garden. And unfortunately, they cross-pollinated. They introduced other genes into it. And so none of the peppers, you can't grow the same peppers again from the seeds because the genes have mixed. They're no longer specialized. Whereas the ones my wife grew on our porch, there were no other peppers growing anywhere nearby, and they've kept their limited gene. Do you follow that? So when you have an isolated population, you lose genetic information. When you have a large population, there's constant mixing, and you get the genes available. So over time, the long-furred canines are less able to adapt, because if you take those ones that have adapted for the northern climate and you take them down to the Sahara Desert, they're always hot because they got too much fur. And so they're not as adaptable as the main dog, which supports both. So the main source of variation that creates species is there's a huge spectrum of genetic information available in most original breeding pairs. In plants, for example, orchids, huge variety in orchids. In the brassicas in plants, all kinds of variety. You think of Brussels sprouts and cauliflower and broccoli, they're all in the brassica family. So there's a big genetic diversity available, or dogs as an example. So in the original equine kind, there are these primary species that have come out. There may be some others, but within these species, there are breeds. I think there's about 700 breeds of horses, if I recall correctly, within the horse species. And these can interbreed, not necessarily successfully producing offspring that can then breed, because they have lost genetic information. So when you breed a horse and a donkey, depending on whether the male horse or female horse, you either get a mule or a hinny, and they are not fertile when you bring them back together because there's been a loss of genetic information over time. Here's where you get an example of the wide spectrum of genetic possibilities in the human genome. Two sets of twins, and this couple, and they're quite colorful. And here are twins that are born, well, came out of the same womb at the same time. And they are, there's quite a spectrum there. And so after the animals came out of the ark, and after the Tower of Babel, the animals started to spread around the new forming continents and around the world, and they became isolated breeding populations. And one of the reasons the isolation occurred was with the Ice Age that developed very quickly after the flood. It created ice barriers that separated breeding populations, and as they went on to islands, and those islands were separated, they became isolated breeding populations, and they didn't interbreed, and over time they lost the genetic information. And similarly, man was driven out. But there's been constant mixing of genes throughout the ages through conquest and wars and all those kinds of things. So human genome is much more continuous than some of the animal genomes are. So when you get a species like this fish, the variety of them, the salmon that can go into different niches. Some can go into only salt water. Some can go into mix of salt and fresh water. Some you find only in fresh water. And this is all available within the salmon genes, and they can specialize into different niches. But there are limits. Salmon don't evolve into catfish or goldfish. There are limits within the spectrum of the gene. But so evolutionists realize this, that what they need is a second mechanism, at least a second mechanism, because natural selection only works on what's available already in the gene. It doesn't give information, it subtracts information. So what they then turn to is that they turn to mutations. But let's take an example. You've probably heard, well, even the SARS-CoV is mutating. You've heard that expression? Like the Delta variant is a mutation. So they turn to mutations as a source of genetic information. So they claim that drug resistance in bacteria is an example of evolution. That's their claim. So and this is taken from UC Berkeley, University of California, Berkeley website. And you'll notice that on the left-hand panel, there's a mix of bacteria there, and the orange one is antibiotic resistant. But it's in the pool already. And then what happens is when they inject the antibiotic in, it kills most of the blue ones, and the ones that survive are the antibiotic-resistant ones. And over time, that's all that's left. As long as the antibiotic remains there, and eventually all of the non-resistant ones are gone, and you've lost genetic information because you don't have the other form. But I want you to notice something that they do here. They talk about it as evolving into the resistant strain. Do you see that? That's not evolution. There's no additional genetic information being added. It's lost its genetic information. This is a constant bait and switch technique that's used in it. No new genetic information. The genetic information was already there. That antibiotic resistant one was in the natural pool. In fact, I'll come to it in a second when I said the fact that I was thinking I had one slide here, but Michigan State University conducted a study on E. coli bacteria, and they ran it through 67,000 generations. They tried various paths. They would use antibiotics, and take through different paths, and they found the same mutations occurring at various times. But after 67,000 generations, it was still E. coli bacteria. Nothing had added, no new genetic information was added. All that happened was that they, at times, depending on what they did to them, injecting antibiotics, they lost genetic information. Never did they increase their genetic information. Now, 67,000 generations with a fairly simple genome. And they're telling us that humans evolved all of this complexity from a pre-hominoid creature in maybe, less than 1,000 generations? And with all of the complexity in the human genome? But antibiotic resistance in bacteria has always been around, at least since Adam's sin. So at the Franklin expedition that around the turn of the, was it the 20th century? Yeah, the turn of the 20th century. That they, or late 19th century, they went to the Antarctica and they died. They got frozen in the ship and they couldn't escape and they ended up dying. And they found them eventually, and they found the antibiotic resistance in those men. So it wasn't evolving from antibiotics, because antibiotics weren't developed for another 70 years or something. And similarly, when you look at isolated tribes that have never had modern medicine, and you take a stool sample from them, and you look at the E. coli or whatever bacteria you're interested in, you find that they're antibiotic resistant. Similarly, when they did it on a soldier that had died during the First World War, and they had a stool sample in the laboratory, and they found the antibiotic resistance there. Bacteria. So they've always been around. It's just that the other bacteria survive better. And so what happens with mutations? Mutations do two things. The main thing they do is a loss of information, or they may have flipped some switches and there's no loss of information, but it's changed some of the nucleotides in the DNA. So it's like a spelling mistake, but the gene can still work, because you can read a sentence with a spelling mistake in it. In fact, you can read a sentence with a lot of spelling mistakes in it. Well, DNA has a lot of error correction techniques, and because we have two sets of chromosomes, if the mother's set is different than the husband's set, you may not get the genetic disease, because you may get the proteins being developed out of the other set of chromosomes. And so, but mutations occur. Mostly it's information loss. Occasionally it's a substitute. And there are other things that I'm not going to go into, but gene insertion and duplication and rearrangement. But none of this is adding new information. It's already taking information and shuffling it around. Because they say, well, look, bacteria can take DNA from other bacteria. But where did that initially DNA come from? They're just sharing DNA. And yes, that can happen. That's insertion. So here is an example. A red cardinal is a natural color. Do you have cardinals here in Alberta? We see quite a few of them. In fact, they're still around in Toronto. They haven't headed south yet. But occasionally, you'll get a mutation, and it's the loss of the ability to produce the red pigments. And you get this nice yellow bird. But if they're in an isolated breeding population, the yellow may come to predominate. If they're in a general pool, often the mutations will get bred out, because they will breed with others that have the broader spectrum. And so, but it's an information loss. This mutation is the inability to produce the red pigment. I mentioned this tea this morning that has been discovered growing in a province in China, and it's caffeine-free. And this tea, notice that it's lost the ability to produce the caffeine synthase enzyme. And the caffeine synthase enzyme appears in chocolate and in coffee and various other substances. It's not just in tea. And the caffeine synthase enzyme produces the caffeine, and the caffeine is an insecticide that protects the plant. And I mentioned that this morning, if you're drinking any caffeinated drink, you're drinking a pesticide. So, but notice what the article in The Economist says. It's lost the ability to produce this enzyme, but it's made its evolutionary journey toward being caffeine-free. Here it is, bait and switch again. This isn't adding information. This is losing information, losing robustness, and they're calling it evolution. Do you follow that? This is what's happening here. Here's another example. Findings which were published in whatever, this Evolutionary Biology, BMC. The black squirrel is actually a gray squirrel with a faulty pigment gene. And it's carried over from the fox squirrel and so on. So when I look out our window, I see all these black and gray squirrels running around. But this is one that has a genetic defect, and it's lost the ability to do it. Now, there's a county north of London, Ontario, where there are white squirrels. They are not albinos. They are white squirrels. And it's interesting that this is another genetic defect from the gray squirrel. And the only reason these squirrels survive is because they're protected by law and humans are taking care of them. It's quite cool in Exeter that they have these white squirrels. But what would happen if they weren't being protected by humans? Think about it. Hawks would spot them instantly, wouldn't they? On the green leaves and on the grass, and you can be sure that they would be snapped up very fast. So this defect would generally be bred out of the population because they would be killed. But because they're protected and people are taking care of them, it's artificial protection in this case. And going back to that tea example, if humans start to cultivate it so that we can have naturally decaffeinated tea, it'll take humans to protect them. They'll have to use other pesticides to protect them because they don't have natural protection. And so we will have to take care of them on that. But this is what mutations generally do. They're detrimental. Cystic fibrosis is an example. Are any of you lactose tolerant? If you are, you actually have a result of a genetic mutation, and it's a defect in your genes. That's, now, not all lactose intolerance. There's also environmental causes sometimes, but it is a genetic defect. And I mentioned previously that there are approximately 6,000 genetic diseases in the human population. So they even if a 1 in a million beneficial mutation were to occur, the likelihood of it being positively selected and improving the species is infinitesimally small, because overwhelmingly mutations are negative or benign. But if the one were to occur, they would be bred out. So I would argue that all mutations are harmful. Now, this is from Nature's magazine on their website. And that you can go through, read page after page after page on, I suppose if you print it out, it'd be a lot of paper, about the detail on mutations. And there's only one example mentioned in passing on this website, which is supposedly a beneficial mutation, and they mention sickle cell anemia. It is claimed that this is a positive mutation. But before I go to sickle cell anemia, I just want to read you at the very bottom of this website. In fact, I think this is the very last sentence or two. It says, together these different classes of mutations and their causes serve to place organisms at risk. Overwhelmingly, the mutations are negative, at risk for disease, and to provide the raw material for evolution. Now watch what they do here. Thus, the mutations are often detrimental to individuals, but they serve to diversify the overall population. So what they're telling you is things that are bad for people, the individuals that kill them, are good for the population as a whole. But there's not a single shred of evidence given on that website. You go and look. There's nothing that supports that statement. It's one of these things, well, we must have beneficial mutations because we see all this variety of life around us. And so they've jumped to a conclusion that is unsupported by a stitch of evidence. So let's look at sickle cell anemia for just a second, because it's the only one that's ever mentioned by evolution as supposedly being a beneficial mutation. It does provide some immunity to malaria, but it's at a very high cost. 25% of the children born who are carriers of this die. That's a pretty high death rate. And are we going to declare a mutation that kills 25% of the children as being a beneficial mutation? But it does provide some immunity to malaria. And what it is it causes it so that the blood cannot carry the oxygen effectively. A good mutation would not impede oxygen distribution in the body. So here's an interesting one. I'm sure we've all heard of this particular SARS-CoV-2 virus. And this was in Popular Science in March of 2020. And they say SARS-CoV-2 is changing at the rate scientists would expect for a coronavirus. It's mutating. Robert Carter, one of our geneticists, in an article he's written on our website, he has predicted that SARS-CoV-2 is going to mutate so much that within about 2 years, it's going to have no damage. It won't have any more damage. Viruses eventually decay. Now, new ones keep coming along, but eventually they decay. And so the mutations here. But Popular Science is telling us that they are mutating, and notice what it says. The bulk of the mutations that appear as a virus spreads are either harmful to the virus itself, meaning it is less likely to survive or replicate, or they don't change how it functions. So we can be happy to hear the SARS-CoV-2 is going to mutate out of effectiveness over time. Right? That's right out of the article back in March last year. You can see the spike proteins there on the outside of the virus, right? So evolutionists speak out of both sides of their mouths. It's contradictory here, because from Popular Science viewpoint, mutations which are deleterious are a good thing, because we want SARS-CoV-2 to disappear, mutate out. But when let's look at genetically modified foods. They do not produce viable offspring. Now, there's people who are against GMO, but most of us probably eat GMO corn, hybrid corn. It basically was grown in Canada because it gives the best yields and needs less fertilizer and so on, but it doesn't grow true. When you plant a hybrid corn, it won't produce the same one that you had before. And so this is genetically modified, but they are less viable than the natural products. Here's an example. The results to date have been reassuring with regard to GMO, that in the genetically altered organisms have proved to be less viable than their wild-type counterparts, and hence unable to affect existing ecosystems substantially. So they're saying that when the mutations occur, they make these organisms less viable. And so if human-directed genetic changes make an entity less viable, unless it's being protected by humans, for example, with fertilizers and pesticides and so on, under what conditions would random genetic changes make the entity more viable? So mutations are always detrimental, or at best they are neutral. There is, other than the sickle cell anemia one, no evolution has ever presented an example of a beneficial mutation. Here's a bacteria that causes stomach ulcers, and it's antibiotic resistant at times because it's lost the ability to produce an enzyme. Here's another bacteria which I'm sure you heard of. It's what causes tuberculosis, and when it becomes antibiotic resistant, it's the ones that have lost or they actually have an enzyme overproduction, and it weakens the bacteria overall. Negative, negative, negative is what we see on these mutations. Here's another example. Genetically altered mosquitoes are less fertile and less healthy than mosquitoes that have not been altered. Mutations, mutations, mutations are negative. And yet these are supposed to be the engine for creating new information. No, never has it been shown, never, that new information is being created. It's a loss of information or a reshuffling of information. And the basic question you have to ask is, okay, there's this genetic information. How did it get there in the first place? Genes don't just suddenly pop into existence. They are complex strings of nucleotides that are built up, and they have a lot of information into them. Every mutation is harmful and causes all kinds of defects. And there's been no example ever of microbes to man evolution taking place beyond, at the very, nothing beyond the family level. Variety occurs within the family level, but never beyond that. Nothing has ever gone beyond. And the family may or may not correspond to the kinds, the biblical kinds. In some cases, it might be the species or it might be between them. But here's an interesting example. These have been isolated breeding populations, llamas and camels, and yet you can interbreed them. So they were originally from a common kind, and they actually do produce fertile offspring. The camas, they'd be actually kind of cute, wouldn't they? But they can produce fertile offspring. So they're not as, even though South America and Asia, they're not as isolated genetically, and they haven't been separated for millions of years, only since the flood. And they still have enough of the overlap in the genetic information that you can breed them. But never has there been a cross-family or cross-kind example demonstrated. And there's no evidence of this type of change, never demonstrated in observational science. Now, I haven't gotten into a lot of detail here at the genetic level, but if you want to explore the building up of what it takes to get to DNA in a cell, a fantastic book that came out last year is called The Stairway to Life. Now, we do happen to carry it on our website, and you can buy it on Amazon or wherever. And it is absolutely mind-blowing. I was speaking in Edmonton at Birch Bay Ranch a year ago, and I read the book on the plane on the flight out, and I just was blown away with it. And I started to reread it when I was flying to Winnipeg last month, and I hadn't quite finished it, so on my flight out to Edmonton, I was reading some of it. I'm reading something else as well, so I was switching back and forth. But notice what it says. From recently acquired insights into the complexity of the simplest organism, Tan and Stadler specify requirements for spontaneous formation of life and evaluate the prospects for natural processes to satisfy these requirements. These two, Tan and Stadler, have amazing academic credentials. They are research scientists. They really know their subject. And the conclusion they come to, there's nothing in this book that's theological per se. It's just science. And they're just saying it's impossible, absolutely impossible, to have the components building up to get to DNA in cells. So let's just talk then. I'm not going to go in. If you want to explore the chemical evolution before we have biological evolution, just read that book. But if you want to take it from there, no matter how much you say that these chemical evolution doesn't occur, evolutionists still insist that it happens. And so one of the ways they propose, because DNA and RNA will destruct very quickly in a water environment, so it couldn't have formed in water, and you need Well, the irony is you need a cell to manufacture DNA, but you need DNA in order for the cell to know what to manufacture. So how it all comes together. But they're trying to come up with a way to have DNA come into existence, and they've suggested one approach is it formed in a clay infrastructure. Another is that molecules have arrived from space. And it actually is given the name interstellar transpermia. They actually have a name for this, but molecules have arrived from space. So, but evolutionists admit that they have no clue how DNA formed. For example, Stephen Hawking. I'm sure you've heard of Stephen Hawking. He had Lou Gehrig's disease, and he's claimed to be this great physicist. And I've read his, well, at least a brief history of time, and I don't find anything in there that's particularly illuminating. But somehow, he says, some atoms came to be arranged in the form of molecules of DNA. We do not know how DNA molecules first appeared. The chances against a DNA molecule arising by random fluctuations are very small. So he admits they don't know, and it's essentially impossible. But somehow it happened. And so he speculates that since we're here, it must have happened, because they won't accept special creation. Here's another example from Scientific American. Unfortunately, there are not enough data to prove or rule out any of these theories, like the mineral structures or molecules from space. When it comes to the origin and early evolution of life, some mysteries seem as intractable as ever. They don't know how DNA could have formed. And they've just outright dismissed the idea that God could have created. And the article concludes, with faith and optimism, that a natural explanation is going to be found. I used this example this morning, but I have to use it again because it's so powerful. Even if we had the basic nucleotides that are strung together to make DNA, even if they could spontaneously form, it's the stringing them together, and the human genome has 3 billion nucleotides. How we're going to string those together. It's not the hardware that's a problem, although it's an impossible thing for it to spontaneously come into existence. It's the software, the information that's built into the DNA. That's the problem. And Paul Davies says, how did nature fabricate the world's first digital information processor? The original living cell from the blind chaos of blundering molecules. How did molecular hardware get to write its own software? I'm a computer programmer. I write Java code, and I've written recently some very sophisticated Java code. And I was thinking in one of my presentations of giving an example. If I just change one semicolon in my program, it blows up. So it's very hard to believe that in DNA, if we don't start mutations, that they're not going to cause damage. Now, God has built all kinds of error correction utilities into DNA. So the complexity of DNA is mind-boggling. Does anybody know what a palindrome is? Do you know what a palindrome is? It's a sentence that can be read in both directions. Madam, I'm Adam. That's what Adam said to Eve when he was introduced. Madam, I'm Adam. That's a palindrome. A more sophisticated one that Napoleon had to voice was, Abel was I ere I saw Elba. Abel was I ere I saw Elba. These are palindromes. I don't know if you're aware, but DNA, remember this is 3 billion nucleotides. DNA can be read in both directions. And it can have different instruction sets. You can start in different places in the DNA and produce different RNA to produce different proteins. You can take different genes working together. There are switches built into what used to be called junk DNA, but now they realize it's regulatory DNA between the genes, and it turns on and off functions. Did I use this example this morning? I can't remember, but epigenetic information is also passed in to cells. And an example is that a woman who is bearing an embryo at high altitudes, the atmospheric pressure information is fed into the developing embryo, and that child is going to have larger lung capacity. That's mind-boggling when you think about it. The DNA can get these signals coming in from outside. And I think I've used this one. I think I used this one this morning. When you cut yourself, no, that's better for cells working together. I won't do that right now. So anything that declares made by God is its DNA. So nevertheless, evolutionists know that DNA, they can't explain how it arrived, but they believe that it must have, and so random chance is going to give them their answer. The Origin of Life Science Foundation offered a $1 million prize to anyone proposing a highly plausible natural process or mechanism for how spontaneously genetic information would come into existence. You'd think a $1 million incentive would be significant. Well, after 13 years, it had never been paid out, and they canceled it. So nobody was willing to step up and say, here's a plausible explanation for how this, even though they could have earned $1 million. Darwin talks about in his book about complex processes, and he says if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modification, my theory would absolutely break down. Well, the Bible tells us that animals were created in mature form. At the molecular level, Michael Behe has shown that there are all kinds of complex machines inside of cells. This is a bacteria flagellum, and it's a motor that spins a propeller. And he argues in there with these components that you can't make them from slight modifications. The whole thing has to be there, all the parts, or it won't work. And he says in his book, Darwin's Black Box, I'm not recommending you buy these books, by the way. Behe is a theistic evolutionist, and he believes in deep time. But what he talks about in micro machines is mind-blowing. But I'm not recommending you get these books. If you search the science literature on evolution, and if you focus your search on the question of how molecular machines, the basis of life inside of the cells they develop, you will find an eerie and complete silence. The complexity of life's foundation has paralyzed science's attempt to account for it. Now, 21 years later, and this is in 2019, he brought out a sequel called Darwin Devolves. And he says the boosters of Darwinism support a theory that labors mightily to explain a crummy 2-amino acid residue disulfide bond, but cannot account for more complex structures. Darwinian mechanism is self-limiting, capable only of eliminating or modifying pre-existing molecular systems. This is the molecular level, and I've been telling you the same thing, mutations at the genetic level. Incapable of building functionally complex structures. If we come up from the biochemical and the biological level of DNA, how do the nucleus of a cell, how did the nucleus that produces the information for proteins, how did that get a nucleus? And then the simplest cell has 482 genes and is vastly more complex than all the components that go into making an automobile, including the software. And how did multicellular entities come? And this is where I use the example of a scratch. Because when you get a scratch and it starts bleeding, the blood begins to coagulate, and the cells talk to one another. They communicate to one another because the cell says, okay, hold on, I'm going to start to seal this and coagulate it. And then once it's coagulated, it now says, okay, I've stopped the bleeding. Now you can start building skin cells again and fill in the wound with skin cells. And then it sheds the scab, and so on. Those are cells talking to one another, working together in that component. Taking it up a level, I mentioned that with bisexual reproduction, you'd get two sets of chromosomes, one from the male and one from the female. How did cells learn to take two sets of chromosomes and cooperate with them? Scientific American has an article that proposes a theory for how the Y chromosomes were developed, because males have a Y chromosome, females only have two X chromosomes. Male have an X and a Y, females have only X chromosomes. So how did this develop? And the article in Scientific American uses words like probably, deduce, seem, and apparently, but it's storytelling. They're not showing observational science. It's all a hypothesis for how it could have developed. And even if a viable life form that had, say, female plumbing were to arrive, at the exact same time, somewhere spontaneously, a male entity would have to arrive with all of the plumbing and and sharing the chromosome sets. This all would have had to have taken place, same time, same place, by chance. Take it up a level here. Convergence is something evolutionists can't explain. Life has supposedly evolved in various different, flight has evolved in various different forms of life. Convergence in eyes. Humans and octopuses and squids have very similar eyes. But the common ancestor that we're supposed to come from had a completely different kind of eye. Hemoglobin is an amazing substance, and it's a complex protein, and it carries oxygen. And yet, it is found in different places in animals, not necessarily in common ancestors, and it's even found in some plants. And how on earth could this complex protein have developed spontaneously multiple times? Well, God used the hemoglobin design multiple times. Another problem for evolution is what's called species stasis. And that is basically that animals don't change into something else. You can breed fruit flies for generation after generation after generation. You can inject things into their environment. They might lose their ability to fly, and they end up having their antennas coming out of their hips and all kinds of weird things. But they never evolve into anything other than fruit flies. They're always fruit flies. Species stasis. Take it up a level to symbiotic relationships. This is what's called a Tasmanian snow skink, and there's the honey bush. When the honey bush is ready to be pollinated, it's got a hard casing on it that only the Tasmanian snow skink opens up so it can be pollinated. And one article said, why did the honey bush evolve such a complicated procedure for its pollination? Hold on a second. Honey bushes don't think. Am I running out of time here? Whoa. Okay. Honey bushes don't think and decide to evolve. It was a random chance. I used this example this morning, so I'm going to skip it. Sociobiology is another problem for biologists because they claim, if you read the literature, everything is a result of our genes. And there's been a book advocating that actually rape is a result of our genes and that rape has a biological purpose. Can you fathom that? And the problem is that if everything is the result of evolution, then all deviances. Look at this article. Prejudices is the result of evolution. So if you're prejudiced, that's okay because it's your evolutionary path. So everything is natural according to evolution. So what evidence exists for evolution? Zero. None. Zilch. Not a single thread of empirical evidence. Nothing supports the claim. Evolution is a theory universally accepted not because it can be proven by logically coherent evidence to be true, but because the only alternative is special creation, is clearly incredible. It's a theory without an empirical foundation, and therefore it is not science. And they have this tendency to rationalize. So why is it so universally accepted? It's because men suppress the truth. Romans 1:18. Most biologists accept evolution as though it were a proven fact, although this conviction rests upon circumstantial evidence. It forms a satisfactory faith on which to base our interpretation of nature. So evolution is believed because it is the main prop for a faith, a religion without God. In fact, this quote here says, the reason we accept evolution is because we cannot allow a divine foot in the door. So, after these presentations, later today, there's a book table over there, and we have various resources. Genetic entropy is a powerful one. Creation.com, if you want, on our website. We have all kinds of good material. We have Evolution's Achilles Heel and an accompanying DVD. Creatures do change, natural selection, but it's not evolution. We also have Creation Magazine, which you can subscribe to, and lots of articles in there. And if you subscribe today, I'll give you a free copy of it and a DVD. The Faith Building Pack is a book for Christianity for Skeptics, is a book on apologetics, and Creation Answers book. And if you haven't signed up for InfoBytes, sign up. Every Friday we send out a mailing with our latest articles and information, and here are some, and there we are. I buzzed through the last few slides.
You guys got a lot of information, haven't you? All right, you store it somewhere. Thank you, Jim. We have let's take about 10-minute break, and we'll come back at 4:45 with our last presentation, all right? 10-minute break, thank you.